All right. Hi, everyone. Good morning. This is our Zarlab Santa Barbara Retreat 2013. Today I'm going to talk about my projects, 3 projects, and the title of the talk is Efficient Design and Analysis of Genome-wide Association Studies. First, I'd like to give an overview of what we're trying to accomplish. Our main goal is to understand the genetic basis of complex traits. And what's a complex trait? These traits, unlike Mendelian traits, have multiple disease genes. When we look at Mendelian traits, we often see a few disease genes are responsible for the disease, and if you have these genes have large effects, and that means if you have these alleles, you are going to have more likely to have the disease. However, for complex traits, we have multiple genes, and these genes or genetic variations have small effects, and not only the genetic architecture, but also the environmental factors and the interaction between the genetics and the environment can induce the disease. So our goal is to understand which genetic variation is associated with traits, and we also are interested in understanding how the roles of genetics versus environment play in a disease. So when we talk about genetic variation, we often use single nucleotide polymorphisms, or SNPs. Here we have 4 individuals, and we have 2 SNP sites. When we compare the genome of these individuals, we are going to see that 99.9% of their genome is going to be identical, except at these polymorphic sites that are conserved in a population. And if you look at these polymorphisms in a population, you are often going to see a complex correlation structure among them, and then this is something that we will use. So what we do is, in genome-wide association studies, we collect these SNPs from individuals who have the disease cases and individuals who do not have the disease, the healthy individuals and the controls. We collect information at each SNP, and then we compare their frequencies between the cases and controls. Here is one SNP site. In the cases, the C allele is in abundance, and in the controls, we have the A allele is in abundance. So it indicates that this SNP site might in fact be associated with the disease, and to assess its significance, we compute an association statistic. And the association statistic we use looks at the difference between the minor allele frequency differences in the cases and in the controls. We normalize this statistic to have a z-score, and under the null distribution where the SNP is not associated, the mean is 0, and we call the mean in this talk as the non-central parameter. And had we known which SNPs are causal, we can also write down the alternate distribution for the causal SNP. And the key message here is that both these distributions have a normal distribution. So we compute these statistics, and we compute a p-value under the null distribution. And here we have a Manhattan plot. On the x-axis, we have the positions on the genome, and the y-axis indicates the minus log 10 p-value, which means higher the value is, the more strongly associated the SNP is. And given a significance threshold value, which is indicated by the dashed red line, the values above the threshold value are the significant associations, and these are the valuable information that we would like to utilize in our research. GWAS approach has been used often in the last decade, and then it led to the discovery of hundreds of disease loci in many complex traits. Here is a catalog of the findings from the recent or older GWAS studies, where we have the diseases as color-coded, and on the bars, these bars represent the chromosomes, and the position on the chromosome for each color represents the findings for each disease. So in outline, first I'm going to talk about a method called Genome-wide rapid association testing, or GRAT. So the problem we are trying to solve in this project is that when we have the GWAS approach applied to a single disease, the computational cost is not very high. However, when we apply the GWAS approach to high throughput phenotypes, such as gene expression studies, we are going to have a computational problem, because we might have millions of SNPs, and if you collect information, gene expression information on tens of thousands of genes, the order of computation is in the order of billions. So what we'd like to do is, instead of testing all the SNPs to identify the significant associations, we propose a 2-stage approach. And in the first stage, we would like to identify the regions that may contain the associations. And in the second stage, we will focus on these regions and only test the SNPs within them. So here's a simple example where let's assume our genome consists of 2 regions, and region 1, it has 7 SNPs, and in region 2, there are 7 SNPs, and the red line indicates the significance threshold. So if we use the current approach where we test all the SNPs, we are going to compute the association statistic on 14 SNPs, and we are going to identify 2 significant associations. But instead of doing this, what we are going to do is, first we are going to assume we are given some proxy SNPs within each region, and these SNPs are going to be best representative of each region. For example, for region 1, a proxy SNP is SNP 4, and in region 2, SNP 9 is the proxy SNP. And to decide which region we would like to focus on, we are going to use a threshold value, indicated as the black line. And what we do is, in the first stage, we first compute the statistic of the proxy SNPs. We observe that in region 1, the observed statistic of the proxy is lower than the threshold, and that means we are not going to test any of the SNPs here. And in the second region, we observe that the proxy SNP statistic exceeds the threshold, and that means we are going to test the SNPs or the remainder SNPs in this region, which we do, and then we will still identify 2 of the significant associations. So this example gives us the intuition that if we choose these proxy SNPs and the threshold values correctly, we can cost the computational cost down and still be able to identify all of the significant associations. However, the problem in the genome is, as I mentioned earlier, we have a complex correlation structure between the SNPs, and to address this, we use a different threshold value per marker. So again, in the first stage, we test our proxy SNP. But now, for each remainder SNP, we are going to use a different threshold value. For SNP 8, the threshold value is indicated as the black line, which exceeds the observed statistic, which means we decide not to test the SNP 8. However, for SNP 10, the threshold value is given as follows, which is which is less strong than the observed statistic of the proxy. That means we are going to test SNP 10. We are given these threshold values, and then we exercise these decision rules, and then we choose which SNPs to test and which SNPs not to test. And at the end, in fact, using the correlation structure information more effectively, we can still identify all the significant associations, and we can further cut the computational cost. So the questions now we are going to address in this talk is, how do we pick these proxies, and how do we pick the threshold values, and more importantly, how can we guarantee that this approach is going to identify all of the significant associations? To develop our method, we are going to introduce 2 metrics. The first is the number of SNPs tested. This is going to be relatively the computational cost. And the second metric we are going to use is the fraction of the significant SNPs we identify. We will call this as the recall rate. If we use the traditional approach where we test all the SNPs, let's assume we have million SNPs. We test all the SNPs, so our NT is going to be 1 million. And since we test all the SNPs, our recall rate is going to be 100%. And let's also assume we are given the proxy SNPs, and 60,000 proxy SNPs, and the threshold values, and we exercise a 2-stage approach. In the first stage, we have a fixed cost of 60,000 because we have to test all the proxy SNPs, and we exercise the decision rules, and let's assume we test an additional 40,000 SNPs. Given that there are million SNPs and we test only 10% of the SNPs, that means we made all the computations 10 times faster. And if these values are selected carefully, our recall rate can be high as such as 99.9%. So our goal in choosing the proxy SNPs and the threshold values is going to be to identify virtually all significant SNPs, to have a very high recall rate, while testing as a few SNPs as possible, where we would like to minimize the NT. The notation I will use throughout the talk is going to be as follows. For the remainder SNP, I will use the subscript I. For the proxy SNP, for this particular remainder SNP, the subscript will be TI. The observed statistic of the proxy SNP is going to be S hat TI, and we will compare it to a threshold value, and we will check if this threshold value is lower than the observed statistic of the proxy. And if it is, we will go and test the we will compute the statistic of the remainder SNP, and then it will be S hat I. And then we will compare it to a significance threshold value T alpha, and then we can deduce whether this remainder SNP is significant or not. However, the NT and RR, recall rate is a function of the particular data, and it doesn't help us on how to choose the proxy SNPs and the threshold values. And we, to address this, we use a generative model for the association statistics, and we look at the expected number of SNPs tested and the expected recall rate of using a particular proxy set and threshold values. And the model we use is, we use the joint distribution of the association statistics of a remainder SNP and its proxy SNP statistic, and we model these distributions under the two cases where the remainder SNP is causal, where it has a non-centrality parameter, and then we can derive the non-centrality parameter of the proxy SNP using indirect association. And the important part is that we can express the covariance between these two statistics, which turns out to be the pairwise correlation coefficient between these two SNPs. And similarly, we can express the joint distribution in the null case, where neither of the SNPs is causal, where the distribution is going to have a mean at 0. To express the joint distribution, we need one bit of additional information, which is to know which distribution we are under. We don't know this, and to construct our framework, we used a probability of remainder SNP being causal. This probability is not known, but we use a uniform distribution for CI, and then we can construct the joint distribution as a Gaussian mixture. Once we have the joint distribution, we can then compute key probabilities, such as the probability of testing a remainder SNP, and also the probability of testing a significant remainder SNP. Using these probabilities, we can then compute the expected number of SNPs tested and the expected recall rate in fixed time for given proxy SNPs and threshold values. If we are given k proxy SNPs in ENT, we are going from the first stage, we have the fixed cost of testing these k proxy SNPs, and from the second stage, we are going to have the expected number of decision rules triggered, which means which corresponds to the number of SNPs we are expecting to test. In the expected recall rate, we approximate it as the ratio of the number of SNPs that we expect to have in our data and the number of SNPs, the significant SNPs we expect to identify in the first stage, and the number of significant SNPs we expect to identify in the second stage. Given these formulation, we can then choose the threshold values for given proxy SNPs, and the formulation is convex, where we try to choose the threshold values to minimize the ENT, such that the expected recall rate satisfies a constraint, satisfies a target recall rate. However, both of these functions are nonlinear, and then we linearize the convex problem to solve it more efficiently. We observe that in the ERR, the part of the function that depends on the threshold values is expressed on the right upper side. We call this function as the expected recall. And one intuition is this function is monotonic in the threshold value. That means we can express all the threshold values as an inverse function, which means we can linearize the constraint, and we can effectively use the Lagrange multipliers and solve this optimization problem very effectively. This plot shows you for two pair of SNPs, one proxy and one causal remainder SNP, we have different values of target recall rates, and we have different values of pairwise correlation between the SNPs. What we try to do is we identify the threshold value such that to check if the observed recall rate will be matching the target recall rate if we use that threshold value, and in the simulations this works perfectly well. However, I showed that for given proxy SNPs, choosing the threshold values is a convex problem. We can solve it very efficiently. However, choosing proxy SNPs and the threshold values is a hard problem. To address this, we use a greedy approach, and we construct a set of proxy SNPs. We start with an empty set, and we try each possible remainder SNP as a proxy SNP, and we compute the performance of using the remainder SNP as a proxy, and then we choose the one that performs well. However, the problem in this approach is that in each stage where we extend the proxy SNP statistic by 1, we have to solve the convex optimization problem for each remainder SNP, and this gives a quadratic runtime. One way to speed up this is to cache the gradient value that we compute in the previous proxy iteration, and instead of computing the derivative or solving the convex optimization problem for each remainder SNP, we use the cached value, and then we compute the performance under that, and we update the gradient after we choose which SNP performs the best at the cached value. Using this approach, we tested its performance in simulated genome-wide association studies. We used real SNP data. We simulated thousands of genome-wide association studies using 1,000 genome data set. In each population, the number of SNPs is indicated in the second column. On average, we have between 6 to 8 million SNPs. And we performed thousands of simulations, and in our simulations, we used 50% power at the causal SNP, which means if we test the causal SNP in these studies, we are going to identify it as significant in half of the time. Our target recall rate was 99.9%, which means we want this function to identify at least 99.9% of the significant associations, and we check what's the speed up. The speed up we gain compared to testing all the SNPs is really good. On average, we have a speed up of 10 times, which means we can perform all these genome-wide association studies 10 times faster than computing all the SNPs. And to give you an idea of how good the recall rate is, in all of these simulations, in total, we had 3.8 million significant SNPs, and we only missed 10,000 of them. We also applied our approach on real data. We used the 80 Asian HapMap individuals that were sequenced as a part of the 1,000 genome project. And after QCing the SNPs and eliminating the rare SNPs, we retained 5.9 million common SNPs. These individuals were phenotyped on 48,000 gene expression probes, which means the order of computation we have to do is 270 billion if we use the traditional approach. We did these computations using PLIG, which took almost 4 months on a single CPU, where obviously we distributed these computations to multiple cores, and we identified 85,000 significant associations. Using GRAD, the proposed two-stage approach, we were able to do all these computations in 35 hours, and we identified all of the significant SNPs. And to give you an idea, in the first stage, GRAD picked 5% of the SNPs as proxies, and on average, in stage 2, it tested an additional 500,000 SNPs, which means the speed up should be 10 times. We also extend GRAD to linear mixed models, and here the problem is if we have certain hidden confounding factors in our association study, such as population structure, this is going to lead to increased false positive rate, and computing the linear mixed model association is computationally expensive as well. One problem of using GRAD in mixed models is that the pairwise SNP correlation, which is the covariance between the statistics, is not going to be the correlation coefficient between the SNP anymore. So to address this problem, we model the distribution of the phenotype. And so in the ordinary least squares approach, we can use the GRAD approach where the covariance is the correlation value. And in the mixed model approach, we have some covariance structure in addition that comes from the hidden confounding. And then we rotate our vector of the SNPs and the phenotype to have independent error, independent residual noise, and we compute the pairwise correlation in this rotated space of the SNPs. We also applied the GRAD on the same data using linear mixed models. To test all the SNPs, we used the method that also came from our lab, MIX, which took almost 380 hours. And by applying GRAD, we were able to do the same computations in 43 hours and get exactly the same results. We also compared the proxy SNPs that we obtained from GRAD to tagging SNPs. We use tagger to this, and the idea is we would like to, given a set of SNPs, you would like to identify the minimum number of SNPs that has a particular correlation, at least a minimum correlation value to the remainder of SNPs. We generated studies using the encode regions from the HapMap data set and for HapMap individuals. In each population, on average, the number of SNPs in the encode regions is 1,000. And when we use GRAD, the number of proxy SNPs was between 55 and 90. And when we use tagger, we observed typically 4 times or 5 times larger number of SNPs to proxy these remainder SNPs. And we would like to mimic the two-stage approach with the tagging SNPs. And what we do is we use a uniform threshold value to choose which of the remainder SNPs to test using the tagging SNPs. For this, we used different threshold values. We started with 10 to the minus 8, and we used lower threshold values such as 10 to the minus 5. And when we used the grad, we were able to identify 99.9% of the significant associations, and we obtained a reduction or a speed up of 10%, 10 times speed up compared to the traditional approach testing all the SNPs. However, with the tagging SNPs, the reduction rate was lower, which it was around 4.5, and you can't put a guarantee on the recall rate, and under different threshold value, you obtain a different recall rate, and you can control this. So in conclusion, we introduced a method to speed up gene expression association studies by a factor of 10, and the key part of this project is that it provides a guarantee to identify all of the significant SNPs with high probability. I also showed that it can be used on top of computational expensive statistics such as linear and mixed models, and an additional speed up can be gained by pre-computing proxy SNPs for certain genotyping arrays or reference data, which means if you use these genotyping arrays, you can perform these association studies using the pre-computed proxy SNPs and solving the convex optimization problem to choose the threshold values for your particular data. And for the reference data, if you impute to these reference data, the approach can be used effectively. So the second paper, the second method I'm going to talk about is RFSS. It's rank-based follow-up SNP selection. And here the idea is we would like to design follow-up association studies to identify all of the significant associations in genome in a particular disease. So when we look at a Manhattan plot, given a significant threshold value, we are going to see that the researchers are typically interested in the significant associations. And if you look under a significantly associated SNP, you're often going to see that since you didn't collect all the SNPs in this data, in this study, you might have some uncertainties about which gene is driving the signal. So here we zoom into a region, and on the y-axis we have the minus log 10 p-value, and on the x-axis we have the SNP positions. The black circles represent the SNPs that we genotyped in our study, and the red circles represent the SNP positions that exist in the database, in a reference database, but we did not collect information on these SNPs. So, and in this region, we have 4 genes, and the significant association is between 2 genes, so it's not clear which of these genes are driving this association or the signal. So what we can do is, one way to solve this dilemma is to genotype these SNPs at these individuals, and then we can observe that in fact gene 3 is driving the signal, which is likely to be the causal SNP in this disease. However, the problem is if we genotype all the SNPs in this region, we are going to have a high cost of collecting or genotyping cost of collecting all the SNPs. And a bigger problem is, had that SNP not being significant but has a strong association, the researcher can completely ignore this region, and had the researcher decided to collect additional SNPs from this region, they would still be able to identify the causal gene, which is not going to be the case if you use the traditional follow-up association study design. So we introduce a method which is powerful and cost-effective in designing follow-up association studies, and the idea is to identify all of the significant candidate SNPs, the SNPs that we didn't collect in the original genome-wide association study that are significant in the disease. To do this, we introduce a prediction model which uses the observed association statistic of the SNPs that we genotype, and we also use the reference information from the reference data, such as the correlation structure between the SNPs and the allele frequencies. So similar to the previous model, in the rank-based follow-up SNP selection, instead of using 2 SNPs, we use the information from the loci, the observed statistics of the SNPs from the loci, and we try to predict for each of the candidate SNP whether it's the probability of being significant or not. And the model is comparable to previously shown method, but however here we have a multivariate normal distribution where instead of 2 SNPs we have multiple SNPs. So using this approach, we simulated association studies where we had in total we had 2 million SNPs, and we assumed we genotype 500,000 SNPs, and we have a budget of collecting an additional 10,000 SNPs to design a follow-up study. So the red vertical line indicates the total number of significant associations that is in the data, which means if we had an oracle which told us which SNPs are in fact significant, we would have a constant line here because we would be picking the significant associations to construct our follow-up association study design. When we used the proposed approach, which is indicated by the blue, we see that it's almost chooses the significantly associated SNPs most of the time. Its predictive power is really high, and then the remainder of the methods are the traditional approaches where we either collect the SNPs that are in the vicinity, or we collect the SNPs that are strongly correlated to the observed significant SNPs. So in conclusion, the framework we propose is powerful and cost-effective to design follow-up association studies, and on average it performs between 20 to 90% better than the traditional approaches. And an important key part in our approach is that it doesn't need the actual genotype data, but we rather use the statistics at the SNPs, and we use the correlation or allele frequencies from a reference data set. Finally, I will talk about a method called HIDE to distribute the heritability estimations, and this project aims to differentiate the effect of genetics and the environment in a disease. Heritability can be, in the narrow sense, can be summarized as the influence, intuitively the influence of genetics in a phenotype, and what we try to do is we try to decompose the phenotypic variation, the observed phenotypic variation, into 2 parts, the genetics, and we assume the genetics is additive, and the environmental factors. So traditionally, heritabilities have been estimated using related individuals, such as the twin studies, and the values obtained from these studies are assumed to be gold standard values. For example, height is a model trait that has been used heritability in a lot, and its heritability has been observed as 80%, blah, 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 which means, okay, we will cut this part. Height has been a model complex trait to study heritability, and through using twin studies, this heritability has been observed as 80%, which means the genetics accounts for 80% of the variation in height in a population. However, we are interested in estimating heritabilities from genome-wide association study data sets where we typically have unrelated individuals. And using the SNPs that we collect in these studies, common SNPs, we would like to infer the genetic similarity and regress it against our observed phenotype. So in the model we have is an infinitesimal model where we have y, the observed phenotype values. We assume there's a constant mean, and then each SNP might have an effect beta I, and we have the residual error E. We assume the residual error is independent and identically distributed, and then we assume the effects of the SNPs come from a normal distribution. If we use the framework, we can construct a linear mixed model where we are going to have the covariates, fixed effect covariates, the genetic effect, and the environmental effect. We can effectively solve this equation and compute the variance explained by the additive genetics and the variance explained by the environmental effects. Furthermore, we are interested in partitioning the heritability into genomic regions such as autosomes or chromosomes. In this model, instead of having one random effect for the whole genome, we are going to have 22 random effects or variance components modeling each autosome. We will refer to this approach as the GCTA approach, and we show that this approach is not scalable and it becomes computationally intractable. Using a reference data or reference GWAS data on height, the Finland birth cohort GWAS data, which consists of 5,000 individuals, we show that if you can't split the autosomes for more than 5 partitions, and the approach fails after this. And to address this problem, where we are interested in partitioning the heritability into higher number of regions, we propose the Heidi approach, where instead of having a variance component for each particular region, we use an alternative model where we have the phenotype and we model the genetic effect of the region we are interested in and aggregate the background genetic effect in a single variance component. It's easier to solve this alternative approach computationally, and we show that in our simulations it also improves the accuracy as well. And the modeling of the variance components as follows: the R, which accounts for the region, has a genetic similarity constructed locally from the region, and for the background effect we represent the genetic similarity or the kinship among the individuals from the rest of the genome. The important advantage of Heidi is that these equations are not related to each other, which means we can parallelize these computations and perform these computations across multiple CPUs. Hence comes the term distributed. However, one problem with this approach is that when we have multiple regions, these regions might be in LD, which means they might share some SNPs, they might share some similar SNPs, and regions that are in LD might take or give heritability or variance explanation to the neighboring regions. And to correct for this inflation or deflation due to LT, we propose a normalization once we compute these, once we solve these equations, which is very simple, simple heuristic. What we do is we first compute the heritability explained by the genome, and then we compute the heritability explained by each chromosome, and we normalize the chromosome estimates such that their sum equals to the genome-wide heritability. We then partition each chromosome into the regions we are interested in, and then we repeat the same process, and now we normalize the region heritabilities to that of the chromosome that they come from. Using this simple approach, we perform simulations using the Northern Finland bird cohort data on height. We used the SNPs obtained from this GWAS data, and we simulated GWAS studies where the heritability is 50%. And I'm going to show now multiple benchmarks where we compare our approach to the traditional approach. And here we have, we split each chromosome into 5 partitions, and the contribution of the heritability from each partition is equal, such that that's why you see the uniform distribution across the partitions. And if you add up the heritability from these regions, you are going to have 50% heritability across the genome. The lines on the above indicates the error, which means ideally we want to have lower error because we know the generating function and we know the true heritability from these partitions, and then we estimate the heritability of each partition under 2 methods, and the method that performs better is the one that has lower mean average error rate. And on average, on equal contribution scenario, our method obtained 8.8% higher accuracy compared to the traditional approach. We repeated this process we repeated this benchmarks under different scenarios, where now instead of having a uniform contribution across these partitions, we have larger contribution from larger chromosomes, and in each chromosome we also have different contribution from the regions. In this scenario we also obtain higher accuracy. And finally we we benchmarked the approaches under a third condition where we have a sparse contribution, and in this case not all the regions contribute to heritability, and in each region only regions 1/3 and the 1/3 and the 5th regions contribute heritability, and the 2nd and the 4th regions in each chromosome has 0 heritability contribution. And in this scenario as well we obtained higher accuracy. So in conclusion, the method we propose performs better than the traditional approach, and we observed 5% better accuracy. And the main advantage is that due to our the power of being able to parallelize these computations, we can compute the heritability contribution from regions that are much smaller than the traditional approach can do, and then we can do this computationally more efficient. In our paper, due to time, we also show, I'm skipping, that we also show that the Heidi approach cannot decompose the effects of population structure and inheritability more accurately compared to the traditional approach. So with I'd like to thank you for your time. And these projects have been a collaboration with my advisor, Eleazar Eskin. And in the second project for follow-up study design, it was a collaboration with Jose Lozano. And the funding for these projects have been from NIH and NSF. Thanks.